All right. Looks like we've got a lot of folks present here today. Thank you so much for joining us. We're nearing 200 people on this call, so thank you so much for being here. Good morning, good afternoon. Welcome to this research webinar, which is being hosted by the Huntington's Disease Society of America and the Huntington Society of Canada. And today we're going to be hearing about the top line results, the preliminary data from the PROOF-HD study of Pridopidine. So I'm Dr. Leora Fox. I'm the Assistant Director of Research and Patient Engagement at HDSA.
And I'm Alexander Maxim, Research Lead at HSC. We'll be introducing our speakers in just a few minutes, but we wanted to first remind the listeners that you can ask questions and let you know about some upcoming events.
So we're going to wait until after the presentation to answer questions, but at any time you can type your questions into the chat box in your Zoom toolbar. We're just using the chat, but it's a closed chat, so only the presenters and the hosts are going to be able to see what you've asked, and we will pose questions. We've got a really big audience today, so we'll do our very best to get to as many questions as possible and then follow up afterwards as needed. I also want to make sure that folks are aware of some upcoming events for both of our organizations. So the 38th Annual HDSA Convention is coming up shortly. We'll be gathering in New Orleans, Louisiana from June 1st to 3rd for lots of educational and research content aimed at HD families. We're expecting up to 1,000 people to join us. You can go to hdsa.org/convention to learn more.
And HSC is beyond excited to share that our 2023 National Conference will be held in person this year in Niagara Falls, Ontario on November 17th and 18th. Registration for this event will open later this month, so be sure to keep an eye out for that.
And I'll also encourage you to check out hdtrialfinder.org to learn more about currently recruiting clinical trials in both the US and Canada. And you can also check out hdsa.org/blog to stay up to date on what's going on with HD research week to week.
We are recording today, so if you want to revisit this presentation or send it to someone else, you can find it at hdsa.org/research-webinars, or you can go to HDSA's YouTube channel at hdsa.org or HSC's YouTube channel at huntingtonsociety.ca using the buttons at the top of our home pages. Usually it's up on YouTube in about a day or two. Now we're very pleased to introduce our speakers. Dr. Michael Hayden is the CEO of Prilenia Therapeutics and a professor at the University of British Columbia. He's well known for his work in HD and other genetic disorders and has published around 900 papers, seen many drugs through approval, including the Korea treatment Austedo, and founded 5 biotech companies, including Prilenia. He's won many prestigious awards, including being named one of the 50 Canadians born in the 20th century who have changed the world. Dr. Hayden has a tremendous impact on the field throughout his work and mentorship.
We are also joined by 3 expert clinician researchers who've been leaders in this trial and in the HD field. Dr. Andrew Fagan is a professor of neurology at NYU. He's been involved in HD clinical research for more than 25 years, has run many clinical trials of treatments for HD and Parkinson's, and has served as the principal investigator of PROOF-HD. He's also served in several leadership roles for HSG, including chair. And in addition to his research activities, Dr. Fagan has a clinical practice caring for patients with HD. Dr. Sandra Kostick completed her MD and PhD at the University of Chicago and her neurology residency at Mass General Hospital. She's been the medical director of the HDSA Center of Excellence at the Ohio State University Wexner Medical Center since 2000, has participated in numerous HSG industry, foundation, and investigator-initiated HD trials, and is dedicated to increasing awareness and education and improving care options for people affected by HD. Dr. Ralph Reilman is the founding director of the George Huntington Institute in Münster, Germany. He's been involved in HD research and care since the 1990s, and through his research in both animal models and in people, he's helped to establish some very important ways to measure health and performance in people with HD that are widely used by doctors and researchers. He also has a strong research interest in novel biomarkers that could be used as measurements in HD trials. So with that intro, I'm going to stop sharing my screen and hand it over to Dr. Hayden to get us started with content.
Thank you, Leora. Thank you, Alex. And just to say, we're delighted to have this opportunity to share some early data on where we are with the proof study. Of course, just to state, only some endpoints have been analyzed. We're very early in the analysis, and we, however, felt it's very important to share with the community what we know today. But this data will change as additional analyses are done, different covariates put into it, and we commit to continue to share data as transparently as possible when it becomes available. This is very much a work in progress. So, of course, this study was designed to look at Pridopidine's outcome on function in HD, measured in numerous ways. These were the PIs that you see here, Ralph and Andrew, and also in Europe, Anne Rosso, who was chair of the EHDN, and Sandra, who's now chair of HSG. So these were highly experienced and expert people to be the PIs in this trial. Let me immediately say, we could not have run this trial without the support of all of you, the physicians, the investigators, the family members, not just the subjects in the trial, but other family members, and we're deeply grateful. We could not do trials without your support, and we couldn't learn to glean more data that hopefully in the long run will have help for the community. So we're deeply grateful for the commitment, and we are, as investigators, inexorably bound to the patients who are playing such a crucial role. Today, we're going to briefly go through some of the data. We want to tell you a little bit about Pridopidine, how it works. I'll do some of that, and then Ralph and Andy will talk about the Proof HD trial, safety and efficacy summary, and then Anne may make some comments, and Sandra may make some comments, and then we'll have chance for Q&A. So we'll go hopefully no more than 30 minutes, maybe 35, to leave time for opportunities for interaction. Just to tell you about Pridopidine. Yes, it's been around a long time. It's a drug that has been involved in clinical trials for Huntington disease, and now has a major feature in ALS, and in ALS, moving towards a phase 3 study in ALS based on the, I'd say, very important findings in terms of progression, speech, and respiration that have been shown in that disease. It's a drug that is safe and tolerable, similar to placebo, which is surprising for a drug that really has impact throughout the brain. It's given twice daily, once in the morning, once in the evening. And we've shown before that this drug may show maintenance of function, motor, and cognition. And this trial was really directed to really see if we can replicate that. The mechanism of action, it binds the sigma-1, and we have some open label data that suggests that there may be some delay in progression that goes up to 5 years, with all the caveats of open label data. How it works, I think we understand a lot now. It's this target is highly expressed in those areas of the brain that are important for HD, the basal ganglia, the cortex. It regulates crucial pathways impaired in HD. It corrects them, it enhances them. And when you lose sigma-1, you get more aggregates, suggesting that loss of sigma-1, and certainly in numerous animal models, if you lose sigma-1, the disease is exaggerated. And this is seen not just in Huntington's, but ALS as well. How it works, it acts on pathways that protect neurons. So it acts through sigma-1, decreases cellular stress and inflammation, and in addition, has numerous protective factors by secreting numerous peptides that are protective, enhances mitochondrial function and respiration. That's so crucial for neuronal integrity. It clears toxic proteins and also enhances connectivity in the brain that often is thought to be a connectivity that may enhance cognition. How it works is that it works on all of these pathways from on the mitochondria, enhancing mitochondrial contact with key organelles, regulating iron homeostasis, stress is decreased, enhances the removal of toxic products, increases BDNF, which is a neurotrope, like a supervitamin for the brain, enhances spines, and improves synaptic function. Now, it works through all of this. The gate for which this pridopidine works opens the door for the sigma-1, but then there are numerous downstream pathways, particularly, for example, in dopamine pathways that are activated by pridopidine, where some of its effects are mediated. So it's the sigma-1 is just the door which opens. Now, the proof trial, and I'll just hand this over to Andy, I think.
Actually, I think Ralph is starting us off.
Ralph, okay, Ralph, sorry.
Right. Yeah, we will, Andy and I will switch back and forth. So hello, everyone from Germany, actually nearly night over here, but it's great to see you all here participating. I just saw more than 230 participants, amazing. So I think that's pretty much the same that we actually experienced when conducting the Proof HD study. You can see a study schematic here. It was pretty remarkable for us, and we are really extremely grateful to all of you, to the participants, to the families supporting this research, because we started this trial. You may remember there was this time of COVID, you remember? So Corona came around, and we were a bit worried whether we would be able to start a huge clinical trial like this. You can see down here that we were aiming at, can you still keep the slide for a second?
Sorry.
We were aiming to recruit 240 subjects on pridopidine and 240 subjects on placebo. And that was really a huge endeavor when starting a trial in the start of Corona. So we were a bit worried. Actually, for the first time, we did some virtual visits as well to make sure that we could get all the data collected that we needed, particularly on the primary endpoints. And we were extremely happy to see that actually this clinical trial recruited faster than we thought. And I think actually I would like to make one statement, being I've been myself in the HSG executive committee for a while, but I'm not as much in the HSG as other participants here are. And I think, you know, it's been great to see the support of HSG coming along that made it possible to run this global trial so well. So next slide will show you what sites were participating. It was really a global trial. So we had about 30 sites in Europe. You can see the different countries here that participated. And on the next slide, you see the sites that participated in the United States and Canada. So again, similar to other projects we've done in Huntington's disease, this clinical trial was run globally and really a very good experience, including retention in the trial. Just about 90% of the people stayed in the trial for the duration of 1 year and 3 months, which is the primary endpoint readout that we will discuss about today. So Andy, you will continue from here.
Yeah, thank you, Ralph. Yeah, before I talk about who we recruited into the trial, I just also want to add my deep gratitude to the patients and families who committed so much time and effort, and who continue to contribute time and effort in participation in the Proof trial and now in the open label extension, and also acknowledge everybody who worked so hard on the trial, all the site staff, investigators, and coordinators, and my colleagues and friends at the Huntington Study Group, and of course my colleagues and friends at the sponsor at Prilenia as well. So the patients that we included in the trial were 25 years or older in age. They had trinucleotide CAG repeat of greater than or equal to 36 and had manifest Huntington's disease. That's what the diagnostic confidence level means. DCL of 4 means people who have clinical manifestations of Huntington's disease and have been diagnosed with clinical Huntington's disease. Had to be an adult onset Huntington's disease, had to have a total functional capacity score of greater than or equal to 7. This TFC score, total functional capacity, goes from 0 to 13, 13 being normal function. And we selected patients who were functioning at above the 7 and above on that scale. We'll show you the scale a little bit later so you can see the kinds of things it assesses ability of a person to stay at work and perform activities of daily living and to manage their finances and to do household chores and to be taken care of at home. There's another scale that measures function in the Unified Huntington's Disease Rating Scale, the UHDRS, called the Independence Scale. And we were looking for people who are scoring 90% or below on the UHDRS Independence Scale. We were looking for people who had a total motor score of greater than or equal to 20. We were allowed people to be on antipsychotic medications, antidepressant medications, anti-chorea medications such as tetrabenazine or deutetrabenazine and other psychoactive medications. The exclusion criteria generally focused on things to make the study safe based on suggestions of issues that might have come up from prior trials. I'm actually not going to go through this in detail because most of these issues didn't turn out to be an issue in the proof trial. We can go to the next slide. These large trials always have to pre-specify primary and key secondary outcome measures or endpoints. And the primary outcome measure for this trial was the change from baseline to week 65 in the total functional capacity. The key secondary endpoints were changed from baseline to week 65 in something called the composite UHDRS. We shortened that to CUHDRS. That includes the total functional capacity, the total motor score, two cognitive tests, the symbol digit modalities test and the Stroop word reading. That combines all of these scores into a single composite score. It's very important that to point out that we pre-specified analyses that would, first of all, we had analyzed the whole group, but we also pre-specified before we did the study that we would analyze the data in people who are not taking neuroleptics and people who are not taking chorea medications. And we'll talk to you about why we did that. In fact, not only did we pre-specify that, but we actually stratified the study by that so that we knew that the people taking those drugs would be balanced between placebo and the study drug. So we could go to the next slide. So just to give you a few definitions of what we mean by primary endpoint and secondary endpoint, primary endpoint is typically an efficacy measure that addresses the main research question. The main research question here was, does the study drug alter the progression of decline in function in patients with Huntington's disease? So we chose the total functional capacity as the primary endpoint. Secondary endpoints are endpoints that support the efficacy as demonstrated by the primary endpoint. And so people can maintain functional status, but we would also want to know, well, do they show improvements in motor function? Do they show improvements in cognitive function or slowing and decline in these measures as a supportive measure for the primary outcome measure? We can go to the next slide. It's also really important when you think about the analysis of these trials is the distinction between a pre-specified versus a post hoc analysis. A lot of data is collected in a bit large trial like this, a huge amount of data, and you could do endless numbers of analyses. So, and just by chance, you might find that some things are statistically significant and other things are not. So it's important to know what was pre-specified, because the pre-specified ones are the things that you say before you look at the data, this is what we're interested in. And so it's a much more kind of unbiased and reliable outcome. And so in this study, we pre-specified, it's really important to emphasize that we pre-specified the analysis not only of the whole group, but of the subgroup not taking neuroleptics and not taking anti-chorea medications. The pre-specified analysis is different from a post hoc analysis where you can kind of just look through the data and look for things that are interesting, look for changes that might be suggestive of something. That can be very helpful as an exploratory type of analysis, but tends to be a little less unbiased and and tends to carry a little less weight than the pre-specified analyses. You can go to the next slide.
Yeah, so I'll take it from here. Why did we pre-specify the analysis excluding neuroleptics and chorea medication? So I want to show you now that we didn't do this out of the blue, but actually data-driven. So you will remember that there was a huge clinical trial, many of you called PRIDE-HD on Pridopidine, where we had about 700 patients and tested different dosages, and actually could conclude very well that the 45 milligram BID dose that we trialed now in PROOF-HD was the best to move forward. And in this trial, in the PRIDE-HD trial, Pridopidine had a stronger effect when neuroleptics were excluded. So you can see here the TFC readout of the total population on the left. That's the change in TFC. There was a benefit in TFC in those people. It was just not significant. It was 0.058, so it scratched significance on the whole group. But then if you look at the right bar, this was in the Pride HD study, in the previous study, those people that were on 45 milligram BID versus placebo without neuroleptics and without chorea medications. And if you look at that, you can actually see that the effect becomes statistically significant and is much bigger. An effect of 0.8, nearly 0.9, that's more than a year of progression in Huntington's disease usually. And that would be something that we would like to get to patients if possible. So note in the Pride HD study, there were about a third of patients that were on neuroleptics. We can keep that in mind for later because we will revisit how this looks like in the PROOF study. On the next slide, I will briefly introduce a measure that we've been using quite a bit in Huntington's disease trials in the last years, including the TRACK HD biomarker study that many of you know. It's the quantitative, the Q motor assessment that kind of translates UHDRS total motor score assessments. Those of you in the clinic and that are on Enroll HD may remember the finger tapping we do, the pronation, supination tapping. These measures really assess fine motor control. You need that for buttoning your shirt, for handling your cup of coffee, and many, many important things. So Q motor was actually developed at Columbia University during my stay when I was there at the HDSA Center of Excellence and under Karen Marder's supervision many years ago to pilot quantitative markers that might not have placebo effects and that are much, much more standardized in clinical ratings across trials. So in this study, we looked at pronation, supination tapping, which you can see here. And if you look at the quantitative objective measure on a force transducer measuring the same thing across the 60 sites, then you can actually see that the lower curve here is the placebo group. Over 1 year of trial duration, there is a decline. That's the progression of the disease. We see that in every study where we use Q motor over years, actually. There is no placebo effect. There's just progression of the disease in the placebo group. But now look at the treatment group. There is a significant benefit at 26 weeks and also at 52 weeks in this measure here in Pride HD and pronation, supination tapping. That's actually the whole group in that study. And now I'll look at the next slide. That shows you how this effect changes. It becomes even better, even more significant if you take off the neuroleptic medication from the Pride HD study again. So this objective data suggested to us that in PROOF HD, we should pre-specify an analysis with and without neuroleptic medication. Next slide, please. The intention being that we didn't want to exclude people with neuroleptics and chorea medication because there's a lot of patients that take it, and we wanted to assure that those patients also have an opportunity to profit from Pridopidine if we see signals on their patient cohorts. So the next slide shows you how many people were actually taking neuroleptics and chorea medications in the PROOF study, the new study we're talking about now. And mind you, it's about 60%, 55% in the placebo group and 60% in the Pridopidine group. I just mentioned earlier that in Pride HD, it was about 37%. So it's a much, much bigger group in this study that was actually on neuroleptics and on chorea medication. I will discuss that in a minute. So Andy, you move forward with the safety data, please.
Yeah, so I'll just present the safety data from the trial, which was very reassuring. So in clinical trials, these double-blind randomized placebo-controlled trials, we don't talk about side effects. We talk about adverse events, kind of adverse things that occur to people during the trial. And the reason we don't talk about side effects is because we can't attribute an effect because we don't know whether people are on the actual drug or on a placebo. We're all blinded to that. So we talk about adverse events and we say treatment emergent adverse events. If the event occurs when the person is taking the study medication, either the drug or the placebo. If it occurs in the screening period or it occurs after study is terminated, the study's over, that's not treatment emergent. So treatment emergent adverse events are adverse events that occur while people are in the active double-blind phase of the trial. And you can see here on the top line that treatment emergent adverse events occurred equally in the pridopidine and the placebo groups, essentially equally, maybe slightly more in the placebo group. We also rate the adverse events according to whether we think, in a blinded way, we think it was related to the study drug. And so AEs that were rated as related to study drug were very closely matched between placebo and pridopidine, essentially the same. Serious AEs that were noted in the trial were pretty closely matched, maybe a little bit more in the pridopidine group, but serious AEs that were rated as related to study drug, there was only one in the whole trial, and that actually happened to occur in the placebo group. In the course of the trial, there were 2 people who passed away, who died. Both were in the pridopidine group. One had a cardiac arrest a year after stopping study medication, so was felt not to have anything to do with the study medication. And another was due to an accident, also rated as unrelated to the study medication. No EKG changes, which is something we monitored closely in either group. This slide shows you the kinds of more common side effects. First of all, more common is maybe not the right word, because even the most common side effects only occurred in 4% of subjects receiving the drugs, and they were equally matched between the study drug and placebo. So in effect, really, this looks like an extremely well-tolerated drug with very little adverse events or side effects. You can go to the next slide. I'll also just present the primary endpoint. So I mentioned to you earlier that the primary endpoint was this measure called the total functional capacity change from baseline to week 65, and in all patients. And so that's the left panel here, shows that the lines are overlapping. So people decline by about a point on their TFC, whether or not they're on the drug or on pridopidine or on placebo, and the lines more or less overlap over the course of 65 weeks. On the right panel, it shows this pre-specified analysis that we said we were going to do in patients who were not taking neuroleptics and not taking chorea medications. And in this slide, this panel, you start to see a bit of a separation. It looks like the people on the pridopidine, the purple line, are remaining pretty flat up to a year, whereas the people on the placebo are declining, up to 65 weeks. It's important to point out that this is not statistically significant, but looks like it's favoring the study drug, pridopidine. You can go to the next slide. Ralph.
Whoops, here we go. Okay, thank you, Andy. Yeah, and you know what, I would just like to say that actually, as an investigator, having been involved in all the pridopidine trials that we did earlier, I was PI of the PRIDE study, and we were involved at the George Huntington Institute in the MBChB trial, and you guys did the HEART study. I was really disappointed not to see the effect on the TFC, but the point is, I think a clinical trial like this, with this huge data set, it needs science behind it. And the science may point us towards very important findings. We've just reviewed the Q-motor results of the PRIDE study, where Q-motor showed a benefit, quantitative measures showed a statistic benefit of the pridopidine 45 BID dose against placebo. And we did these Q-motor assessments, pronation, supination, tapping, finger tapping, again in the PROOF HD study. Let's look at the results. So if you look at the pronation supination test in the whole population, so that includes the people on neuroleptics, that's what you see. Again, the blue line depicts the placebo group. There's a decline in function and motor function and fine motor control as predicted. That's what we measure in every Huntington group. But we see again a benefit here, which is significant, and even in this total group at 26 weeks. And there is no significance in the rest of the 2 assessment points, but there is still a clear trend, I would say. And now move to the next slide, please, which shows us the Q-motor finding and pronation, supination, tapping without neuroleptics and chorea meets. And you see all of a sudden there are significant findings, very robust significance in 26 weeks, but also maintained significance, very good signal at 65 weeks. So I think this tells us again that here clearly there is an effect of pridopidine in the brain. The pridopidine compound modulates fine motor control. It's something important for you in everyday life. And on the next slide, you will see it's actually corroborated by the finger tapping as well. You know, if you manipulate needles or button your shirt, this is important to have fine motor control, actually maybe more important than involuntary movements, as we can see here. So very robust and significant findings. The Q motor doesn't go up and down. There's not much variability. We see that in pretty much every study. And so I think this is a clear signal that pridopidine conveys an effect on motor features in the Huntington patients treated with it. And, you know, I mean, we have other diseases where some drugs work in this population, some drugs work in the other population, Parkinson's disease, other diseases. Not always everybody is treated with the same drug. So if there is a subgroup of patients profiting from pridopidine, I think given the fact that we don't have options to treat Huntington's disease yet very well, except for reducing the chorea, I think, you know, it would be really important to note what this means and do further research as to whether a significant subgroup of people may profit from it, maybe also earlier people that are less treated with neuroleptics or with chorea reducing medication. So a very clear and robust reproduction of the findings we saw in the Pride HD study. So the second big study showing an effect like this. Interestingly, there is the measure cUHDRS, the Composite Unified Huntington's Disease Rating Scale that Andy mentioned before as one of the secondary endpoints, the key secondary endpoint, actually. So this is a measure that's being used more and more in the HD community. It's a composite. It combines function, motor, the total motor score, and cognitive endpoints. So if you look at this measure, we also did it in the Proof HD study. On the next slide, you can actually see that the Q motor significant findings, the changes over time in the study in the Proof HD study over 1 year and 3 months, they correlate to the cUHDRS, meaning if you have more profit in the Q motor findings, you also have a better performance in the cUHDRS. And Andy is now going to show you how the cUHDRS performed in the Proof HD study, the secondary measure.
Yeah, so the next slide. Yeah, so kind of like the TFC on the left, if you include all patients, the composite cUHDRS declines over the course of the 65 weeks of the study and is really similar between the pridopidine and the placebo group. But similar to, actually much more, I think, impressively so than the TFC. Now, when you just look at people who are not taking neuroleptics and not taking anti-chorea medications, there's clearly a highly statistically significant benefit to being on pridopidine. The purple line shows improvement and sustained benefit up to at least 52 weeks, whereas the placebo group is declining in this population. And it's statistically, as I say, statistically significant up to 52 weeks. And we can go to the next slide. And then finally, for this cognitive measure, this measure that's been used very, very frequently and validated in many, many studies in Huntington's disease, is a reflection of cognitive ability in people with Huntington's disease called the Stroop word reading, or SWR. We'll look at that. Again, on the left, very similar types of findings as what we saw from all the other measures, from the Q motor and from the composite UHDRS. It looks like if you include all patients, here there's a little bit of a benefit, but it's not statistically significant. If you exclude on the right panel here, if you exclude people on neuroleptics and anti-chorea medications, all of a sudden this becomes highly statistically significant up to a year, and impressive separation between the study drug on the purple line and the placebo in the blue line. Again, reflecting pretty much the same observations we saw with all of the other pre-specified outcome measures. So we can go to the next slide.
Yeah, so I will take the first summary slide. So I think I would just acknowledge again that clearly, and we've shown that here, the primary endpoint showed no benefit from pridopidine in the overall group. But I think sometimes things aren't all that straight. You have to look into things a bit more in detail and look at the science. It is really, I think, compelling evidence there that the pre-specified analysis that we performed, excluding neuroleptics and chorea medications, and all of us clinicians know that neuroleptics and chorea medications do have side effects, and they have impact on things like cognition, on fine motor function and stuff like that. So if you exclude those, the overall cUHDRS progression that assesses, as I alluded to earlier, multiple areas, multiple domains of Huntington's disease, shows some interesting clinical reads Q motor has a robust signal for pridopidine to have an effect on the nervous system, reproducing previous findings from the PRIDE-HD study. So there is objective data of this to work in the brain. And it was indeed, I think, interesting for us to see that cognition had a positive effect here as well. So the not only trends, but actually significant findings observed across all Q motor measures. Actually, in this trial, all Q motor measures change was a much bigger group than in the PRIDE-HD study. I think this is really interesting, not placebo effect affected evidence for a central effect of pridopidine here. Additional analyses are ongoing. I think it is warranted to look carefully at all the signals we've seen here, again, to see whether maybe a significant subgroup of Huntington patients could not profit from this. And so I think the open label study actually is a very important thing for us as well, to have the data on it and to get further knowledge about it. Pridopidine is certainly a very safe and well-tolerated drug. And for that reason, I think if it conveys benefits, we would really like to explore whether they can be brought to the patient community. Michael, you will summarize the rest.
Okay, so I think what is encouraging is that overall, this is the first drug in the history of Huntington disease that has ever shown improvements in delaying progression as measured by cUHDRS, motor function, and cognition. So these are important findings that certainly give us hope. We also see that cUHDRS, which has been published now and recognized in Europe, not fully recognized in the US yet, but likely to, that cUHDRS represents the most sensitive measure. And it's important to note that at 1 year for each of these, cognition, motor, and cUHDRS, these were the same as baseline. There was no progression, at least held for 1 year. So we need to continue to watch these and see what happens over time. The Q motor was useful and indicated improvement in patients on neuroleptics and chorea meds, but it was further improved when those meds were less present. We still have to look at numerous things, when the patient was on the measure, what the dosage was. We haven't done any of that. Important to note, this replicates. This is now replication of PRIDE. And I do want to mention about the open label. That is so important. I mention about the open label because there was a drug just approved this week that failed its primary, but in fact was approved based on a biomarker and also some evidence for open label extension. Our open label extension will provide continuous access to the safe drug. For some, we think it will provide overall positive benefit while being safe and well tolerated. And of course, if you stay in the OLE, you may continue to derive this benefit. Importantly, in our partnership with the community, this will also contribute key data, which will provide information on long-term efficacy and safety. And for some who have been on placebo and then switched to pridopidine, we will be able to see whether you derive benefit from that. That's a crossover design in a sense, and we'll learn a lot. The OLE will continue for about a year, but this does not prevent you from participating in the other studies later. And in addition, we're considering ways to make the OLE less burdensome, less frequent visits, somewhat decreased assessments, so this places less burden on the families and the community themselves. Now, I'm left again to thank everybody. This is such a partnership with patients and families. We share the same hope. We work together in an effort to find something that can be of helpfulness for patients with this disease. We see, for example, just this week, beautiful data in Alzheimer's that has taken a long time to get there, many failed trials. And so we're learning, and hopefully the data that we have here will allow us to be able to bring this drug to families where appropriate. We thank all the sites and the investigators. The Huntington Study Group has been a very valuable partner in this. We have other partners, which are TFS, CTC, EHDN, Prilenia, a tiny company with, for most of the time, less than 25 people essentially coordinating 2 trials, one phase 2 for ALS, another phase 3 for Huntington's, and have worked far beyond what would be expected. And then our sacred relationship with all the organizations. We're grateful to the Huntington Disease Society of America, which I've had contact with since I first met Marjorie Guthrie over 45 years ago. The Huntington Disease Association, Help for HD, the European Huntington Association, Huntington Society of Canada, celebrating its 50th year this year, HDO, various country organizations, Holland, Italy, Germany, Spain, Scotland, and of course, HD Reach. So we're very grateful, and Germany, I didn't mention Germany. And I know, I just want to say that I know that Sandra is also here. She may have some comments, and we very much welcome an opportunity to have discussions with you and continue to have ongoing discussions.
So much, Dr. Hayden. We're very appreciative that all of you took the time to present for the North American community today and for the European community. I know that you jumped off a webinar with them to come to us. So thank you very much. We've got a lot of great questions coming in, and I want to start with one about neuroleptics, because as family-facing organizations, we are sort of hearing talk and concerns around the idea that families are going to want to know whether it makes sense for them to come off of neuroleptics, or to understand sort of what the implications are here for neuroleptics. And so one question that I do see in the chat here is how current neuroleptics, for example, tetrabenazine, Austedo, how do those medications compare in terms of affecting some of these measures, for example, Q motor? And so does Pridopidine in that sense perform as well as some of the existing drugs that are available to treat people with HD?
Let me just answer, and then Ralph, I'm sure, and Andy will answer. So firstly, this analysis is still ongoing. Nobody is advising anybody to go off any medications at this point. We do not have the data on which to say that. And certainly they treat different things, just like, so the tetrabenazine and Austedo, the drug that I took to approval, treats chorea. Pridopidine does not treat chorea. Pridopidine is not an anti-chorea medication. If the patient desperately has chorea that needs treated, the patient should be treated appropriately. There have been trials to show Pridopidine, to show tetrabenazine and Austedo improve chorea. There are other studies that show whilst they may improve chorea, they may not be quite as useful in other domains, but I don't want to get into that now. There are no long-term studies on neuroleptics and their role on Huntington disease, but they do play a role in providing symptomatic relief to patients. And at this stage, there is no reason to change anything. We want the patients who are on these drugs to continue. At the end of all of this, when we have a full analysis, perhaps totally outside of Pridopidine, because we have a whole placebo group that's been on this for a long period of time, prospectively, we will be able to comment on what happened to these patients on placebo and chorea and neuroleptic medication. We are not there yet. So right now, no change. Stay as is. If a patient needs the meds, please stay on them. And in the end of all of this, we'll be able to come to some conclusions about pattern of practice in a study that just on its own really adds huge value just with regard to the use of neuroleptics and chorea, but we're not ready to make any announcements at this very early stage.
Yeah, maybe I make a comment just to say that formally Q motor hasn't been used to analyze tetrabenazine or ditetrabenazine. There was actually a consideration to use it in the ditetrabenazine trial, but at that point it was decided not to do it. So we don't have data on that, but certainly I have to say that the data that we have generated now very much encourages us to possibly look at that, for instance, in the setting of Enroll HD or other studies, right? And maybe Sandra, you want to make a comment about the global thing. We haven't had you speaking yet, so go ahead, please.
And Ralph, just one point, we will be able to answer that in this study because we have placebo just on chorea and we'll be able to look at it, but we haven't done it yet.
Yeah.
I was expecting the question to come up from our community is, does this mean that the Korea medicines are bad or the neuroleptic medicines are bad? Should I go off them? And I've been thinking about this as we've been discussing the data. And I think as a community and as a practitioner and people prescribing the different medicines, and particularly the people who put so much effort into this study, we put so much effort, years of effort into the trial. We're just beginning to analyze the data. And as Dr. Hayden pointed out, especially with the crossover and the open label, we're getting more data. And I think it's too early to say, and make any definite opinions, but these medicines are also being prescribed for a valid reason. And that I think that it's best both for the study and for us to learn more as an HD community. We volunteered to participate in this and do this, to kind of wait a bit before jumping to conclusions. We've got a lot of hints here that are very promising, and we can continue as a community to help with learning if we continue the way things have been for a bit longer.
So thank you very much. One of the questions that I've seen a lot of people writing have been about the open label phase. I was wondering how much longer will the open label phase run, and when will participants be told their group assignments, whether it be Pridopidine or placebo, during the double-blind phase?
Well, thank you, Alex. And we've learned a lot from the ALS trials because, for example, Amylyx, as well as tofersen, got approval based in part on the success of the OLE trial. And tofersen showed some improvement late in some of the measures, and Amylyx too showed some further improvement. So at the moment, we haven't got a decision when we're going to let people know, but it's soon. We have to come together to decide when we will unblind these patients. But it is most important. The OLE will last, at the moment, it's committed to last to next February. That would mean that we'll get a lot of data to see how people do on and off, with and without neuroleptics, with and without chorea, staying on the meds they were on, for us to really be able to answer. If the drug works and the patient's been on placebo and then goes on to Pridopidine, we should see some, and if it works, we should see some benefit. We'll watch those slopes very carefully.
I would like to make one comment, maybe, Michael, if I can jump in here. I think we've actually seen this in a previous clinical trial. I mentioned earlier that the Q-motor measures, which you've seen earlier, which provided the evidence in Pride and proof for efficacy of Pridopidine, they don't have a placebo effect. If you don't have a placebo effect, you can actually measure against the baseline. And that means we can possibly, that's my speculation, see that those people who are switched from placebo to Pridopidine in the open label extension might see a similar effect than we have seen it initially in the Proof HD and Pride study. So that's interesting data for us to have at hand, with Michael referring to the fact that some of the drugs have gotten approval or conditional approval now on biomarker level. Q-motor actually is not a biomarker, it's a clinical measure. And if we would see effects that are reproduced across multiple cohorts and time points, that effects are there on the motor domain, this may give us a window of opportunity to get the drug maybe to at least to a subset of people. We'll have to see.
That's why, and also its safety and tolerability is so important. There are no adverse events, and being in the open label will not prevent you from being in another trial in the future. For this trial, there is no limit to what other trials you can be in. As you know, other trials with ASOs, if you're in one ASO trial, it's difficult to get into another one. For this trial, at the end of it, of open label, you can choose to go, if you want, into another trial, and there's no issue. The half-life of our drug is 8 hours. So small washout to be able to do that. But we would encourage you to stay at least for a year, so we can get that data that'll give additional data on the potential therapeutic benefit in the long term.
We've got lots of great questions here. I want to jump to one about TFC, one of the primary endpoints. So the question is really about the range of people in the study, and whether it's fair to assume that, most of the people who are taking medications for chorea, for example, are sort of more progressed in their disease stages, and whether that was separated out during this study. So yeah, someone's asking about the distribution of that TFC.
That's a great question, and we don't have the answer to that yet. It's
Okay.
something we deeply looking at. We would expect, similar to the person asking the question, that these would be later patients, in the TFCs of 7 to 9 maybe, or even lower. But we don't have that yet. And in particular, that would be for patients who started these drugs during the trial. And there were quite a few patients. Also, by the way, even though neuroleptics were balanced at baseline, the patients on active drug had much more chorea medication than the others. So they ended up, there wasn't totally balanced. For example, 24% of the active group later, there were 22% who had chorea at baseline, and the end 28% compared to 19% on active. So we also have to correct for that. None of these corrections have been modified. So the groups were balanced for neuroleptics, but not balanced for chorea meds.
Can I make a clarification? That's something I've seen a number of people asking in the chat about what are neuroleptics and which ones are the chorea medicines, because not everybody seems to understand that, and there was some confusion. So the chorea medicines, the two that are FDA approved in the United States, are Xenazine, which is also tetrabenazine, and Austedo, which is deutetrabenazine. So those are the two chorea medicines. The neuroleptics that are used in the United States are for chorea and other problems in Huntington's are risperidone, olanzapine, Abilify, haloperidol, and a few of the newer injectable medicines. So those are just to clarify, because there seemed like there was a fair amount of confusion in the chat about the difference between a neuroleptic and an anti-chorea medicine.
And let me just make one point. You may wonder, why do neuroleptics somehow mask the effects of pridopidine? Pridopidine acts on the sigma-1 receptor, is an agonist. Haldol, for example, not a commonly used one, is a sigma-1 receptor antagonist. So we're quite interested also to look at them and say, okay, if they're at baseline on Haldol, probably the door is not available for pridopidine to work. But what happens if pridopidine is used first and Haldol comes later? Do you then not see the effect? We're busy looking at all of that to see timing, and that may also be important. It's also well published, and the HSG has published this with Carl Kiebitz and Andy. You've also been part of this, showing that tetrabenazine, wasn't Austedo, had great impact on chorea, but its impact on functional capacity was negative. That's already published. There's lots of data published that you can affect one symptom, but make another symptom worse. And that data is out there for tetrabenazine and others. And so it's very much a physician's decision as to what is most important for the patient. And this comes back to, but it's helpful to have all the knowledge that is there. But there's lots of published papers that look at these drugs and show the impact in certain symptoms and then show how progression and cognition is impaired. So these are not surprising in terms of when we really reflect on the detailed literature that goes back some way.
Thank you very much. Given the data you presented today, were there any plans for a future study of pridopidine on pre-symptomatic patients?
Well, that's a great question, Alex. Of course, this drug would be best, for patients who don't need neuroleptics and chorea meds, although Qmoda is still good even with those. So, and a study in early patients, maybe prodromal, is something we're thinking about. But the first step is so we're not there yet, but it is there for us as we think about who would be best for this patient. And it's a big population, particularly if you can have neuroprotective effects without any adverse effects of any other drugs. So this is a brilliant question that we haven't yet fully embraced. I think we need to go through the results of this study, see the groups that were on meds. We don't quite know enough to know whether the people on meds were later. One would expect that, but we'll have the data and we'll sort through all of this. The next step, of course, is deciding whether we believe there's enough data. Just to, again, say, tofersen is a great example, but just one. 10% of all drugs approved by the FDA since 2018 missed their primary endpoint. And they were approved based on secondary, post hoc, and historical data. I'm not saying we will or we won't, but I am saying there's good precedent for this, both in Europe and the US, and we have to decide at what point we might want to have discussions where appropriate as the data evolves.
I would like to make one quick comment though, Michael. I think, honestly, as a researcher being involved in Huntington's disease for so long, I would really love to do a first clinical, or there was one trial, a smaller trial in the past, but do a real clinical trial with a medication, with a drug in prodromal pre-manifest Huntington's disease. And with the Q-motor data we received from PROVE and PRIDE, Q-motor actually worked up to 2 decades before onset of the disease in track HD. So we would even have a clinical measure to go for, even if regulatory agencies don't accept that yet. I think the more evidence we collect with a safe drug like Pridopidine, orally taken, you don't have to do any injections, you don't see any worsening as we saw it in other treatments, I think it would be compelling to try it one day.
I would love to, too, Ralph. We just have to make sure we have resources and the ability to do it. But so one step at a time. But yes, I'm fully in agreement in principle with you.
I want to be mindful that we've got lots of questions and that we're coming to the end of our hour here. So maybe we'll pose one more, and then if you're able to stay on for another couple of minutes, we can do another. But yeah, there's a couple of questions in the chat here about this effect that is sort of a positive effect for about a year, kind of a balloon mid-study. But there was a significant decline in that second year, right, to the point that the placebo and Pridopidine groups kind of met back at the end of this designed study. So people are asking sort of why would that be for 1 year, and whether there's intention to kind of explore that a little bit more.
So I take a quick hit on that. I mean, on Q-motor, that was not the case. So the quantitative measures actually were a little bit better towards the end. And by the way, we didn't go into that in detail yet, but the primary endpoint was at 1 year and 3 months, but we still have data extending beyond that from part of the people. This will all be data we will be looking at in the next weeks, because this is just a few days after that database lock. We share all these thinking with you, and there may be very important findings also then, particularly from the open label extension study, because if that gives us long-term data on quantitative unbiased measures and we have a good readout there, clinical measures have placebo responses, and we have to look at how the different features of the cUHDRS behave in detail to make a more informed decision about that.
And it's important to understand, so what is the data shown? So firstly, it shows improvement until about 1 year for most measures. Q-motor continues to 65 weeks, no decrease at 65 weeks. What we also see at 65 weeks so far is it's not significant, but there is still a delta between placebo and group that shows a trend to a better response. We are looking at 78 weeks. We have data that's coming out, and then, of course, the long-term data. So I think it's too early to comment. What we can say for sure is that patients actually have improvement in the first year, and we'll have to see how long this is persistent. And that's why we really ask patients to stay in the open label so we can get more data that looks at this from 78 weeks and then further, which will give us more information. It's still very early disease does progress. So the other point is this does not halt progression of disease. It may modify the course, it may slow progression, it may improve cognition, but over time we would expect this to still get worse. But look at Alzheimer's disease. Look at the big announcements this week, 27% decrease in progression. We've actually shown improvement, not decrease in progression. Look at the one from Lilly today, 33%. It means the slope goes down, but it's a little less slope. In ALS-FRS, there is no drug that keeps it at baseline. It slows the slope. Here, for the first time in a neurodegenerative disease, even for 1 year, we actually see improvement. That's not been seen in other neurodegenerative diseases. So we've got a lot of work to do. Let's learn what we have so far. Let's look at the long-term effects. 78 weeks, hopefully we'll have, maybe even in time for the HDSA meeting coming up in June, where we can start giving more data about that issue. Of course, it's an issue that we want to watch very carefully.
And Michael, I might just add, just to clarify that, about 3/4 of the patients in the trial went to 78 weeks under the blind, and it was a pre-specified outcome measure endpoint to measure the difference at 78 weeks. So yes, we'll be very interested in seeing that.
Yeah.
I want to pose one final question, I think, just because there are a couple of things related to access here. So first, will the open label be open to additional participants beyond those who participated in the Proof HD trial? And in general, what does this data mean in terms of, if you could comment on access or on compassionate use, because we always get questions from the community about that.
Well, let me just comment. Limor, are you here? No. Okay. So let me comment on open label. So the open label is currently scheduled to go till February next year. Our plan is to continue to do that. During this period, there's also been an expanded access program for the sites where investigators were. We had a number of 30 or 40 patients. I know in the ALS trial, we had 5 expanded access programs, all run through Mass General with a few other sites. So we are looking to, that expanded access program is not full, and people should go to their own sites where the study continued, and there would be availability as part of expanded access. After this, we really haven't made decisions as to what happens in February 24. But you know, what we're doing, for example, in the ALS trial, is we've now, Mass General has applied for our drug to have expanded access as part of the ALS Act, where we can give 200 patients access to the drug for at least 2 years, supported by the federal government. We'll see how that application goes. That's just gone in early May. We may consider doing a similar program for access for Huntington disease patients. So the goal would be, as we learn more, and if we continue to show improvement and benefit, we would look to expanded access, but we don't have plans at this point, besides the sites where there is an expanded access. I don't know, Yael, are you on the line? No. Okay. My Israeli colleagues, it's already getting a little late at night. Yeah.
Well, I was going to say, don't forget, Europe is dark meanwhile.
Yeah.
Yes.
Okay.
Well, given that it's a few minutes after the hour, I just want to thank all of our speakers very much again for making the time to present really to the global community today in your many presentations. And we look forward to your presentation at HDSA's convention. And to all of the participants today, I want to thank you all for coming. I really feel that it's an active advocacy to engage with research and to continue listening as all this clinical trial news comes out. So thank you everyone for being here, and we'll see you at the next webinar. Have a great weekend all.
And just on behalf of everybody, Leora, so beautifully arranged together with Alex. We just want to thank you for making this available and doing it in such a professional and easy way for all of us. We appreciate all of this and very much compliment HDSA and HSC for the professionalism in which this has been done. So thank you all and have a great weekend.
Yeah, thank you very
For the
much.
opportunity.
Very much appreciate
Bye-bye.
it. Take
Take
care.
care.
Thanks, everyone.
Bye-bye.
Bye.